Bismillah Rahman Rahim. Welcome back, friends. Welcome back to another video lecture by Kami Microbiologist. Today in this video lecture we are going to talk about self-splicing process of RNA, that how it will occur and what are the main things, what are the introduction and what we required in self-splicing. In previous video lecture we talked about the spliceosome mediated splicing, which are actually mentioned in the video of splicing technique. So we already talked about the splicing technique, the overview, and before that we talked about the 5' capping, 3' polyadenylation, and we talked about the post transcriptional modification as well. So if you missed this video lecture, it is present into my channel. Go back and watch it. So let me erase this one portion and let's start the self-splicing technique. Okay, so let me write mediated splicing, or we call that self-splicing. Okay, now first of all let me talk about the introduction of the self-splicing or intron mediated splicing. Then we will talk about the requirements, and then we will talk the mechanism as well. What is intron mediated or self-mediated splicing? As the name suggests, self-mediated splicing, it is the process of the RNA splicing in which the intron, which are the non-coding region which don't take a part in the protein synthesis, are cleaved out without the involvement of any kind of proteins and some other SNR molecules. And due to this way, when the intron are removed out and then axon portion are rejoined together, this process is called as self-splicing or intron mediated splicing. So in this case we don't require any kind of the protein. So if we if you guys remember, we talked in previous video lecture in spliceosome mediated splicing, we talked that we need a SNRP, SNRP, and we need 150 proteins and small nuclear RNA as well. So these things we need in the spliceosome mediated splicing. But in this case, we don't need any kind of the protein, and we don't need any kind of the small nuclear RNA. So simple, what actually we need in this case. So let me write here that what actually we required in this case. So let's suppose this is the RNA, premature RNA. They have the exon portion, as you can see, and they have also the intron portion. So let's suppose this is representing the intron portion. So all of you better know intron are in a large amount while exon are in a low amount. Means intron are 97% while exon are the 3%. Okay. So among this is the intron. You know what actually we have between this intron and exon, there is a specific nucleotide sequences, and there is a specific site which we call that 5' splice site, which is present at that specific area. Okay, so we already talked about that 5' splice site, which is present at that area. Then at that specific area we have the 3' splice site, 3' splice site. So remember, this is the 5' and this is the 3' area of the premature RNA, and this is the pre-mRNA. Now this area is called a 5' exon, and this one exon is called as 3' exon. Now this 5' exon have 5 phosphate group, and they have also 3' hydroxyl group, and they have also 5' phosphate group and 3' hydroxyl group as well. Now in this intron portion, this green one, this green line represents the intron portion. So remember, between or in the mid area, or not in the mid area, somewhere in the mid area of the intron, we have a nucleotide which we call that adenine. So adenine will be present at that specific point. And in adenine nucleotide, they have hydroxyl group which is present at the 2' sites. So if we see the nucleotide structure, so I already talked the nucleotide structure, but let's suppose this is the nucleotide structure, as you can see. Okay, now this is the carbon number 1, 2, 3, 4 and 5. This is oxygen, so phosphate, and there is also phosphate, and it will be connected another nucleotide, then another. Okay, so this is the ribonucleotide, let's suppose, and here we have the hydroxyl group, and this is the base. Any kind of base will be connected here. It could be a guanine, cytosine, thymine. Let's suppose in this case we have the adenine base are connected here. So let me write adenine. Now this 2' hydroxyl group we have at that specific area. So let me draw 2' hydroxyl group is also present. So in this case, this is the adenine which is present in, not in the mid area, but somewhere in the mid area of this intron. And moreover, we also need some of the nucleotide which is present in the exon as well as in the intron. So in the exon we have the nucleotide like so we have intron we have the nucleotide like A, G and GU. So remember, in this case, A, G are present in the exon portion, while GU are present in the intron portion. And between this G of the exon as well as this G of the intron, between them the 5' splice sites are present. Okay. Similarly, in this case, we have the adenine, guanine, and guanine uracil. So in this case, the nucleotide are reverse or opposite, as you can see. So adenine and guanine are present in the intron portion, while guanine and uracil are present in the exon portion. And between the guanine of the intron and guanine of the exon, we have the 3' splice sites. So these are the main things that we required in the self-splicing. Okay, and this adenine, as you can see, this is called as branch site. So let me write, this is called as branch site, and from there the splicing or start. So we can say it is the start point of the splicing. Okay, so what this is the main things that we require in intron mediated splicing. Okay. Now what happened actually during intron mediated splicing? First of all, this 2' hydroxyl, as you can see, adenine, they have lone pair. So this lone pair will attack to this phosphate which is present in the intron portion. Okay, this in the intron portion there is a 5' site. So in the 5' site we have the phosphate group. So this phosphate group is attacked by this hydroxyl group having the lone pair, and we call that nucleophilic attack. So it will attack to this one area. And after that, what we get a labyrinth-like structure. So let me draw here. Due to this way, the exon will be separated. So as you can see, so the next we have the structure like that. So the exon will be removed out. And this is the intron portion, as you can see. So this is the phosphate group, and this is the hydroxyl group which is present with the of the adenine, and both attack to each other. That's why it make a labyrinth-like structure. And this is the second exon, as you can see. Okay, now the first exon are cleaved out. This is the second exon. After that, what happened? With this first exon, we have also the hydroxyl group present. 3' hydroxyl group is also present. So they have also a lone pair of electron. It will attack to this phosphate. alpha phosphate of this 5' area. Then next what we get the structure like that. So this is the first exon having the hydroxyl group, and this is the second exon having the phosphate group. So what happened once the nucleophilic attack, this is called nucleophilic attack. So let me write nucleophilic attack. Due to this nucleophilic attack, what happened? The two exons are connected to each other and the intron portion are cleaved out. So this is the intron portion which make a lariat like structure. So this intron portion are cleaved out and we get the exon, and after that these two exons are connected to each other. First exon and this is the second exon, and then it leaves the nucleus, and once it leaves the nucleus then it reach to the cytosol of the cell or in the cytoplasm. Then after that it is ready to produce the protein. So what we get? Ultimately we get the product protein. Okay, so this is how the intron mediated splicing will occur, or self splicing. So again in this case we don't require any kind of the protein, but just we require the nucleophilic attack or hydroxyl group in both intron and exon. Due to this way the intron portion are cleaved out automatically and the exon portion are connected to each other without the involvement of any kind of the protein. So finally we get the mature mRNA. So this is the mature mRNA. So all of you better know this mature mRNA will give rise to the protein. So this is all about the intron mediated splicing or self splicing. I hope you will get idea about the intron mediated splicing or self splicing. But if still you have any kind of question, you can write it into a comment. I will give you answer as soon as possible. If you like this video, make sure to hit the like button, share this video, and definitely subscribe the channel to get more interesting videos like that. And stay tuned, because in the next video we are going to talk about alternative splicing in a detail. Thank you so much for watching.